The ubiquitous nature of memory is so pervasive that it is often overlooked that everything we do involves the use of our memory. In work or school, we equate memory with learning information for a test or a project. Yet, our memory constantly details our experience, whether we consciously make an effort or not. It provides the basis of selfhood through our autobiographical memory, the stories and details of our past, the ability to manipulate information through accessing past experience to make sense of new information, and the ability to learn skills that require an implicit knowledge and memory to refine and perfect. As a simple example, we just displayed an image that, when prompted, you will likely recall and likely be able to describe in relatively precise detail. Similarly, if we showed you various images, you would likely be able to identify which image we first displayed. The question that underlies the crux of memory research is what mechanisms allow this information to persist in our brain. How do you know that you've seen this image before, and how can you describe it? What biologically occurs for you to retain this information over time? There are different aspects of memory whereby mechanisms in the brain will allow for the short-term retention of information and experience, as well as the long-term retention of information. The systems that allow this are distinct, yet the biological mechanisms that facilitate the persistence of this knowledge over time, be it a short time or a long time, are now only being pieced together. There are generic terms most people are familiar with now, such as brain plasticity, that convey an adaptability necessary for memory formation. Yet this only suggests there is an ability for the brain to modify itself, without specifics as to how. Memory is a very complex topic. However, understanding some basic mechanisms may illuminate more than just whether we have seen this image before, but also why and how you are able to know this.
Change takes place at the level of the synapse through experience. Specifically, there can be greater facilitation for presynaptic neurotransmitter release and an increase in the number of postsynaptic membrane receptors. This changes the neural circuitry by altering not only the strength, but also the number of synaptic connections. Cells that fire together wire together. Donald Hebb proposed an explanation for neuronal adaptations during the learning processes. It describes a basic mechanism for synaptic plasticity, wherein an increase in synaptic efficacy arises from the presynaptic cell's repeated stimulation of the postsynaptic cell. Although traditional models of synaptic plasticity explain the brain modifications at the molecular level, the neural mechanisms of long-term memories require alterations of proteins that control gene expression at the level of cell-wide transcription. The interface between environmental factors and gene expression in the context of memory formation has been until recently unclear, since mRNA and proteins are not sufficiently stable to explain the persistence necessary for long-term memory.
A mechanism referred to as epigenetics influences the way our genetic code is read without affecting the original code itself, like the way punctuation marks can influence the meaning of a sentence without changing the words. There are genes that promote memory formation and others that suppress it. When an interesting or repeated experience occurs, memory-promoting genes are activated, and the suppressor genes are inactivated by epigenetic mechanisms. A common type of epigenetic modification is called DNA methylation. Methylation involves attaching small molecules called methyl groups to segments of DNA. When methyl groups are added to a particular gene, that gene is turned off or silenced, and therefore no protein is produced from that gene. Through the action of DNA methylation, the genes that suppress memory are inactivated, thereby potentiating memory formation. Histones can also be modified epigenetically. Histones are protein components of the chromatin, around which the DNA winds for tight packaging. With tightly packed chromatin, DNA is less accessible to the transcriptional machinery. One of the epigenetic processes that involve histone modification is through acetylation. This describes a process that can modulate the accessibility of chromatin for transcription, which can lead to an increased expression of memory promoter genes. These changes are influenced by various environmental factors and experiences. And for this reason, the stability of these mechanisms provide a plausible model to explain how memory may persist over a lifetime.
In summary, epigenetic mechanisms promote the formation and storage of memory at a cellular level. Through experience, environmental factors interact with genes through epigenetics to modulate the formation and consolidation of memories. In other words, the reason why you would remember the picture of the chimp amongst a display of other images is because epigenetic mechanisms provide a means for fast-acting memory formation. These mechanisms also explain stable and long-lasting memory imprints. Synaptic plasticity is localized at the level of the synapse, while the impact of epigenetics is cell-wide. So the next time you see the image of this ape, remember how chimply marvelous your mind is.